Okay, so it's 7:30 and we're just going to get started. Good evening, everyone. Thank you for joining us today for Science Cafe with Jigesh Shah and Jaya Vatsyayan. Before we begin, I want to take a moment to thank the Cary Library Foundation, whose mission is to raise private funds for the library. Their support enables us to bring programs like tonight's event to you. The foundation plays a unique role with the Science Cafe series and also produces the content for these programs. Jigesh Shah will be your host for the evening. Jigesh is a distinguished scientist with a background in biology, computation, and engineering, who is also a Lexington resident, former Cary Library Foundation board member, and the creator of the Science Cafe series. Jigesh, welcome. And I am now going to hand the evening off to you to host and introduce our special guest for the evening, Dr. Jaya Vatsyayan.
Hi, thanks, Helen. Thank you, everyone, for attending today's Science Cafe. I'm Jigesh Shah, as Helen described, local Lexingtonian scientist, and most importantly, host for this evening. So for those of you new to the Science Cafe, we gather regularly to meet the scientists that are in our neighborhoods, which, as many of you know, not too difficult to do in Lexington. So we endeavor to learn kind of how they got to where they are, get an idea of what they do, and maybe even how they do it. So before we start this evening, I wanted to let you know that we'll be taking questions from the audience through the Q&A function of Zoom, and then we'll cover them after we have our discussion with our guest. So with that, let's get started. This evening, we're honored to have Dr. Jaya Vatsyayan joining us as our guest. Jaya is a member of the Cary Library Foundation, a Lexingtonian, and most relevant for this evening, the director of medical writing at Crispr Therapeutics, a gene therapy biotechnology company based in Cambridge. We've called this evening Bench to Bedside. This is a term often used in academia, industry, and regulatory agencies to describe how the development of medicine starts as an early scientific activity, goes through some kind of discovery process, and then ultimately develops into a medicine that will treat patients at the bedside. And as we'll see this evening, Jaya is an exemplar practitioner of this. So thanks for being here, Jaya. And let's get started and at the beginning. So actually your beginning. So tell us a little bit about, where you grew up, how you got into science.
Thank you, Dr. Jigesh. I'll call you Jigesh. It's a pleasure to be here. It's an honor. Well, to answer your question, I grew up in North India in an eastern state called Bihar, which is almost like 800 miles from India's capital, New Delhi. And then I went to college there and later on moved to West India and then New Delhi and then abroad.
Yes, yeah. So we'll get to some of that. So when was kind of the earliest sense that you had an interest in science?
Oh, that's an interesting story. Well, as a teenager, I wanted to be in theater. There was a short moment of time where I was interested in drama and dance and wanted to be in theater. The traditional path to college in India during my high school days was medicine, engineering, commerce, business. And you had to take this all India entrance exam and state level exams to get into these courses. But the decision had to be made very early. Like in the sophomore year of school, you had to decide what courses you would select in the junior year of high school. And at that time, since many of my family members were doctors, it just influenced my early interest in the medicine program. But during that process, I realized that I found organic chemistry, genetics, and biochemistry very, very interesting. And then later on, I went to college, to science college. And I think my introduction to biotechnology was because the professors had organized a workshop, which was like path to careers in biotechnology, and which motivated me to pursue research in that area. And incidentally, the government of India had taken a lot of interest in biotechnology. And while I was still in the middle school in 1986, they had established a Department of Biotechnology, a department that was actually responsible for administering development and commercialization in the field of modern biology and biotechnology. And when I completed my undergrad, during the completion of my undergrad, when this workshop was organized and we were introduced to biotechnology, we came to know about this all India level entrance exam that could actually offer you the provision of internship and scholarship to learn these great modern molecular biotechniques like PCR and gene sequencing and like very prestigious research institutes, which actually these research institutes are all under the canopy of Department of Biotechnology of Government of India and have actually laid the foundation for the biotech revolution that has occurred in India in the past 20 years. Like the National Institute of Immunology, the International Center for Genetic Engineering and Biotechnology, Center for Molecular Biology, and Tata Institute of Fundamental Research, Indian Institute of Sciences. These are these research institutes that during my time in 90s offered like all these opportunities to learn molecular biotechniques and enhance your skills in PCR and gene sequencing.
Yeah, so driven by the government. So from the government, India decided that they would, create this workforce, as it were, to be able to establish that. How is that, how has that played out in India?
Yeah, well, yes, the purpose was to prepare a workforce. I mean, for the upcoming biotech industry, for researching biodiversity, traditional medicine, food, plant, and animal biotech. And these endeavors did add to the advancement of biotech. And, I think the country is being considered as one of the top destinations for investment. In fact, in the past 20 years, India has been able to build this platform for vaccine manufacturing. And as a result, you see that AstraZeneca's COVID-19 vaccine is produced by Serum Institute in Pune. And biotech sector is actually considered key contributor to India's economic growth vision. So although I left India in 1990s, but yes, India was preparing for that.
Yeah, so that's pretty exciting. I think that there's also certainly the sense that a number of the South Asian diaspora, well trained in India, then moves to many parts of the world, and I think you and I represent part of that as well. So yeah, so that's exciting. So it's not just homegrown Indian biotechnology activity now, really being able to seed activities around the world. So
Absolutely.
Very exciting. So that actually formulated your early interest in biotechnology as a distinct path from medicine.
Right.
And one of the questions I always ask our guests is, so most of the if there's any kids on, or if there's parents, you get them to go to college, and then you decide to go back to college after you're finished. But you didn't go right away, right? You actually did something before you went back to college for graduate school.
Yes. Well, during my internship at National Institute of Immunology, I was, I think the Biotech Consortium of the Government of India was trying to prepare us for the industry. So I gave it a pause and thought I should enhance my molecular biotechniques and perhaps just pause and think about going abroad for a PhD. So I joined a virology lab at National Institute of Immunology, and which specialized in Japanese encephalitis virus and dengue. And there was a project that was actually trying to develop a PCR-based diagnostic test for Japanese encephalitis virus. And this virus is carried by mosquitoes and is endemic in certain parts of India, and children are vulnerable to it, and there are deaths that occur because of it. So the diagnostic test was being developed, and now actually there is a vaccine also in India for it. So that allowed me to become a PCR specialist, and also gave me some time to think about going abroad for PhD.
Must have, yeah. So it sounds like you went through a time where current events must seem deja vu at some level, and maybe we could talk about that. But I think that it's remarkable that every one of these kind of endemic viruses do go through this path of ultimately a PCR diagnostic test and then the vaccine. So Yeah, so it's played out many times. I would say that, this is on a much larger scale, but yeah, it must have been a great viewpoint from you to be able to see that happen.
Yes.
So, so then you finish your PhD, and then you decide at that point to move on for further training. And so often there'll be something called a postdoctoral fellowship. And where did you do that? And then what did you do afterwards?
Yeah, so actually, for my PhD, interestingly, I ended up in Taiwan. And yeah, and it was because my family was there at that time. I decided I had a 4-month-old baby, and I decided to join them. When I ended up there, I discovered myself in this science city called Hsinchu. And it has 2 prestigious universities. And I was like, okay, let me see what I can do here. And I started exploring, and I found that the National Tsinghua University had a very good life science department, where they had Institute of Molecular Medicine, which was basically, you learn a lot about opportunistic pathogens, about gene regulation, regulation of gene expression, and mostly biotech world. And I applied as a PCR specialist, and I joined what would be my future lab, my future PhD supervisor. And I started also helping people, in my lab mates with manuscript preparations. So I was doing, working as a PCR specialist and helping them with manuscript preparation. And that ultimately, I realized that it was a world-class university. So I ended up taking the local entrance exam and all the stuff that, the criteria that needed to be fulfilled. And I enrolled in year 2000 for a PhD program. And started working. My dissertation was on transcriptional regulation of human gene encoding UDP glucose dehydrogenase. The Human Genome Project was going on at that time, and it started in 1990 and ended in 2003, and created this huge database of human genes. And this particular gene was encoding for an enzyme that catalyzed a reaction that led to a product which was a crucial part of cell signaling in neurodegenerative diseases and cancer, et cetera, et cetera. But ultimately, I was able to complete my PhD, publish, and I was invited to be a research scholar at University of Pittsburgh Medical Center, the Hillman Cancer Center, where I joined an NIH fellowship to work on post-transcriptional modification of proteins that are involved in cancer pathways.
Yeah, so that's a remarkable time to do your PhD while all of this information is unfolding about the human genome. And I think those of us that kind of were training during that time, it just the amount of information that was coming was remarkable. So it's an exciting time to get your PhD, and then I have to say that you get your PhD, you finish your postdoctoral work. So at this point you are full-fledged scientist, and then you have to kind of decide what you want to do next. And I mean, it does feel like at least I can say for myself as a scientist, you often are asking yourself what you're going to do when you grow up multiple times during your life, because there are these break points where you finish training and you're doing the next thing. So what was that like for you in terms of that next step once you've become a full-fledged member of the scientific community? You have to make another decision.
Right. Yeah. I was like, okay, should I just stay in the academic setting and get this grant and the next one and the next one, or look at the big picture of translational science. So I think the one thing I keep telling my kids is that serendipity is something that often happens in your life, and it is a phenomenon that the dictionary defines as finding valuable or agreeable things that you are not seeking for. And this is exactly what happened with me. And, I was in India, I was trained as a molecular biologist for the biotech industry. And then I spent 10 years in academic setting, in a very monofocused bench side scientist scenario. And then I told myself, after attended a few conferences and some brainstorming sessions, etc., I ended up working at the Cystic Fibrosis Foundation at Children's Hospital for University of Pittsburgh Medical Center, where I worked very closely in a lab that was working very closely with for clinical trials in young patients with cystic fibrosis. And I found clinical science appealing to me. I started exploring beyond that monofocus. And so when the opportunity came to work at Vertex Pharmaceuticals as a clinical scientific writer in cystic fibrosis, I did not miss that chance. And they were in the midst of the first groundbreaking cystic fibrosis medicine, which, as ivacaftor, Kalydeco, it was the first medicine for underlying cause of cystic fibrosis mutation in the CFTR gene. And I told myself, okay, scientific and manuscript writing skills, working almost 24/7 in the lab, and then project management skills, probably all these could come handy in my new profession in the industry. And so I made that move.
Yeah, that's exciting. I think I want to dig into, at least maybe define for the audience, this term that we use very easily is translational science. Yeah. And it is remarkable that kind of what we're doing is two areas of medicine and biology that, strictly speaking, like you might think that they have very similar languages, but they don't, right? Mm-hmm. And you took your training in biology, deep understanding at very molecular level, and decided that what you wanted to do was apply that to medicine and kind of clinical need. And so, as you said, there were a number of skills that you found translating, kind of easily into this new position. But what else, what did you find more difficult in terms of making that move to both industry as well as into this translational clinical role?
Well, I think the most difficult thing was most of the transferable skills I was carrying to the industry, right? I think I was carrying, working hard, effective written communication, and knowing your audience, and complex data interpretation, etc., time management. But what I had to, the learning curve was like knowing all these guidelines around good clinical practices, around global document submission harmonization process, which is called the International Council of Harmonization, has laid out this broad process where you have to know how to prepare scientific packages for review by various regulatory agencies. And then, many regulatory guidelines that during the interaction with the sponsor, FDA, EMA, they keep coming up with new guidelines because they learn from the sponsors, across the sponsors, they're working from across the globe, and they're coming up with these new guidances and guidelines. So you have to keep up to date with those. And because your scientific data is written within that guideline, which is very different from what you do in the lab. Like in the lab, I wrote my experiments, I wrote my interpretation, sent to one journal. The journal would say, oh, no, do more experiments, and get back to me with this data in order to justify this. And that was looked like my work, you were more, but here you had to let go and see the big picture in terms of regulatory guidelines.
Yeah, and I think I would imagine that kind of as many scientists that transition into these roles, the connection between what they do at the bench and how it gets turned into a medicine is kind of a remarkable journey. And it's not something that, we often teach our students and mentees. And so I think that they're often quite surprised, by how much work there is. But, I think that this is a great point for us to kind of really maybe dig into kind of what is that path. We all hear about these remarkable scientific discoveries. We'll talk about some of them this evening. But then what is the path, kind of what is the science journey that then somehow allows us to build and really use this in human beings? Yeah. So you learned all of this work as you went along. So maybe you could take us through that journey, because, kind of I'll speak for myself, the idiot's guide to kind of what we would call, drug development.
Right. Yeah. For example, if you look at the, I'll take an example of the cystic fibrosis drug, right? So, scientists discovered, oh, this is a misfolded protein that actually gets sent for degradation. How do we take it up to the cell surface to make it work like a chloride channel? And they found a drug that could target and chaperone it from the endoplasmic reticulum all the way up to the cell surface. So that was a discovery that happened in the lab, after many, many screening of the drugs, of the compound that could do it most efficiently. And then after that discovery, you start with this, you start testing it and you've tested in all these cells, then you test it in animals. And once you are very confident that, yes, I can now reach out to the regulators and tell them about my study design, about my preclinical study design. And that is called the pre-IND, which is like the step before you start your application for investigation of new drug, because then your discovery becomes a new investigational drug, like investigation new drug, which is called IND. But then before that, it's called the pre-IND stage, where you prepare this scientific document, you write up all your data of your manufacturing, that is how you chemically synthesize that drug. And then you move to, okay, how you tested in vitro in cells, how you tested in vivo in animals, and what are your plans for your first in human. And you start interacting with the regulatory agencies like FDA, and you share your information in something called a briefing book, and with all the associated documents that go in with it. And then the FDA reviews it, and they come back to you. They set up a meeting. They send you a written response, and they provide feedback on that design. And they say, oh, okay, your design looks good. However, I've these feedback based on, because FDA has its own set of scientists who are reviewing your work. And these scientists are looking at several other companies that are trying to develop, either working in that particular therapeutic area or working in that particular drug design or that indication. And they provide the feedback, and they say, oh yeah, now you're ready to file your investigation new drug, and you can start your clinical trial, first trial in humans. And that is for safety, followed by efficacy. And you do pharmacokinetic and pharmacodynamic analysis, where you see how your drug breaks down in the body and how that breakdown product can do to your body. So after all these studies, you reach to a pivotal stage where you do a pivotal study design, and that is in a larger sample size, which is larger study population. And it has particular kind of statistic analysis, which has to meet certain endpoints. And those are all, through all these phases, you continuously interact with the FDA and scientific advisors across Europe and Australia and wherever you want to file that drug in the market. And after all these interactions, at every end of phase, whether it's phase 1 or phase 2, there's interaction with the agency where they give you feedback. If this drug is in a population where it's like an orphan drug, that means it's in such a small population, such an acute unmet need that there's no other medicine, then FDA and regulatory agencies have certain way of accelerating that review. That means they have more interaction with you. They are able to give you more, frequent feedback. You're open to setting up more meetings with them, and they prioritize the review. So there are a lot of these designations that the regulatory agencies assign to the drug development based on the need, the unmet need, the rarity of the disease, unmet need, and obviously how debilitating it is. So ultimately it goes through all these phases, and once it has reached pivotal and meets all the endpoints, it goes for pre-market approval discussions with the agency. Again, every stage you have to prepare your data package and share with the regulatory agencies. And when the pre-market approval time comes, again, you ask them the questions, okay, this is our study design, this is what we have obtained. Do you agree that we are ready for registration? That means we can file for the application, the new drug application for approval in the market. And once that is approved, you prepare these 1,000 pages of data that you have to do in a very structured and very following all the regulatory guidelines. You have to prepare in various modules for efficacy, safety, PK, and for not only for clinical, non-clinical, and also for chemistry, manufacturing, control, because that is such an important part. All these are important parts of a drug approval. And finally, that 1,000 pages goes for review, and ultimately there's back and forth, there's advisory committee, there's lots of questions, and you have to provide responses over the weekend. So like within 2 days, whatnot, and ultimately a drug gets into the market after thorough scrutiny. And that's just one regulatory agency. There's Health Canada.
Right.
Yeah.
So I every single one of these regulatory agencies is really looking at your scientific, at the very beginning, even before you get into people, to look at the rationale by which you believe you can treat this disease with your entity.
Yes.
And it's really, here's the rationale, here's the basis for why we think it's going to be safe in people. Why? Because, we all should be very clear, like all of the preclinical animals that we use really don't tell us what happens in a person.
Yes.
Yeah, and that's I think the biggest step that oftentimes, is really important to remember that, we know not nothing, but probably less than we think we do before we get into that first human being.
Exactly, yeah. And that is why most of the drugs fail in the first phase itself, because what you're learning from the disease models in animals are not truly representative of what would happen in the humans. That is why you start with the smaller population, you test for safety first, you test for PK first, and then you move to testing for efficacy.
Right. And so then, once you get there, as you pointed out, you build a clinical, you build a design for a clinical trial. Mm-hmm. And this is a terminology that, is quite, kind of part of our zeitgeist now. We're constantly waiting for all of these vaccines to get approved. And you talked about accelerated. So there is this, for the, everybody must be familiar with the, EUAs, emergency use authorization for, some of the vaccines that we've seen. So, we're all getting inundated by this, the kind of work that you do, on a daily basis now. But navigating that is really your role, because the interaction with the regulatory agency is through these written documents.
Right. Yes.
Right. Yeah. So although they're structured, it is still, at least in part, understanding the science and how to structure it. I would imagine that there's a lot of people to contribute to this as well. So that's yet another skill that you have to acquire.
Yeah, soft skill for navigating through these cross-functional team members, the statisticians, the clinical pharmacologists, the physicians, the regulatory, like the chemistry, manufacturing, regulatory people, all sorts of, so the whole team is comprised of subject matter experts in their own area who just come together, and you have to really navigate through their expertise and bring together a document that is well crafted, consistent, and communicates clear message. So for that, the project management piece is very important, where you have to lay out a very effective process of getting this document from scratch to completion.
Yeah, so that's kind of as a background here. So now let's put on top of this new era of medicines that they're not the pills, they're not the maybe the injections of antibodies that people are getting, gene therapy. So maybe you could just give us kind of a quick description of how you think about what a gene therapy is.
Well, I would say that gene therapy is not it's something that scientists have been trying for decades, like more than 50 years, because the genetic inherited disease is so debilitating that the idea did not escape the scientist's mind. How can we treat diseases at the genetic level? And what that meant was that, you do a technique that modifies a person's gene to treat or cure diseases. And the way this technique would work is that you replace a disease-causing gene with a healthy copy, or you inactivate a disease-causing gene that is not functioning properly, or you introduce a new or modified gene. But to introduce this into the human cells, you needed a way to take it into the human cells, right? Deliver it. So then that discovery of the delivery tool has been a journey of greater than 50 years, I would say. and some of that tool has been used in the COVID-19 vaccine that we have seen, like the viral vectors where the spike protein DNA was put in and introduced into the cells, into the humans. And I think so that's why it's been a long journey to reach here. And all that has been done through gene therapy, discovery of various delivery tools, discovery of how a disease-causing gene can be replaced or inactivated. All this has also enhanced the other area of science. And obviously, this all started with the characterization of DNA by, we all know Watson Crick, Franklin Wilkins, and then also knowing how messenger RNA was formed from DNA, how the genetic discovery of genetic code, the RNA, how RNA is translated into protein. But the very important genetic engineering technique that was important for laying down the foundation of this idea of how to add a corrected gene into cell was when the scientists were able to develop a molecular tool by inserting a DNA into a DNA molecule called plasmid that was discovered in bacteria. And when they were able to transform the bacteria with this inserted gene into this vehicle called plasmid, they suddenly realized they could also do it in humans, and that's how the whole idea started. Then the additional discoveries were made, how the catalytic, RNA has catalytic properties, and it can splice itself also. The splicing mechanism of RNA, which is the I would say at the foundation of the medicine Spinraza that was approved for spinal muscular atrophy. But the splicing error is corrected. And then, so all these biotech revolutions and the Nobel Prizes, like literally every discovery was won a Nobel Prize. All this laid the foundation for for gene therapy. And I think the first gene therapy trial was in 1990s. A lot of lessons learned through the 90s, and FDA and NIH came up with the consortium that closely monitored all the gene therapy trials. Also, the scientific community further focused on improved and safe delivery tools and expedited safety reporting and enhanced molecular tools. And all this led to the initiation of the gene therapy trials we saw in the 2000s of these genetic mutations, also for infectious diseases such as HIV, for inherited blindness, for spinal muscular atrophy, for hemoglobinopathies, and also, cancer. We have way to go for cancer, but yes, we have some cellular gene therapies that were approved for cancer as the third line therapy, but still in patients that had no other treatment available. So this renaissance of gene therapy, basically from 2016, we've seen so many of these approved, like at least at least 6 or 7 approval. Especially remarkable is the in vivo, gene therapy in retina, like the Lexterna and then Zolgensma, which is for SMA. And these have also laid the path for therapies that could treat diverse clinical indications and tissue targets and offer even like broader impact and foundation for many conditions, many genetic conditions that are still being explored. Like every year, I think the 2021 FDA received 900 INDs for gene therapy. So there's a lot to be done, but still a little, some milestones have been achieved.
Yeah. So I think that one of the things that you said that I think is worth just reiterating is, kind of the natural language of the cell is this genome. Is it DNA, RNA, and the idea of being able to create a therapy that has the same language as what the cell does is really not a new insight. But the biggest problem was how to get those instructions into enough cells that you could generate kind of a therapy. And I think this is what's often called the delivery problem. Kind of we know what gene is. We've known for many years genes like cystic fibrosis, sickle cell disease. We know what the mutation is, and we know the pattern of inheritance, but we haven't been able to kind of put the gene back in. Yes. So now, as you point out, viral vectors, lipid nanoparticles, these are all things that are being added back in. And those are, I would say, one class of drugs where therapies are the DNA or the RNA that gives the instructions. I think, probably the newest revolution, and as you pointed out, a very recent Nobel Prize was given in the context of CRISPR. And this is an area that you work on now. And maybe you can tell us a little bit about how that's distinguished from kind of the gene therapies where you put in the instructions. What is CRISPR, and what is it? How is it different than the kind of the traditional gene therapies?
Well, the conditional, just as you mentioned, Jagesh, the conditional therapies are, you add a corrected gene, you replace, or, gene replacement or gene augmentation by putting into a vehicle and introducing. But you don't have much control over it. You don't know where the gene will go and do whatever, right? And you can only work on like a single gene. But what the next generation gene therapy is allowing us to do is to edit precisely, like, across multiple loci and across, I would say it's still to be explored, but, you can actually delete, insert, or disrupt harmful genes, repair mutated genes using a very specific RNA and an associated nuclease. And this is, there were other nucleases also, like for HIV gene therapy, TALEN and zinc finger nucleases were used, but this is more specific because this is more, I would say, a very programmable gene editing tool because it just needs to change the sequence of the RNA. And by changing the sequence, it can find its target that it can pair with, and it can take the nucleus along with it to cut that particular target. So that leads to more precise and more accurate gene editing, and it also leads. It can also not only just add, it can delete, insert, disrupt.
So it's a finer tool in some respects, right? In the other one, in the other gene therapies, we do have like maybe kind of instructions. Where you're taking the entire instruction and you're putting it in. And so it's telling you what to do. But here it's taking the instructions that are already in your cell and then modifying them in situ, like in that space. So it's a remarkable, finding, right? This the idea that this a therapy and it's fair to say that this is what the discovery is how old, of the actual enzyme itself, CRISPR. Cas9. How old is that discovery?
Well, in 2007 or 2008, I think what we call the yogurt bacteria discovery.
We can go through that. But I'm like, but now you work at a company which actually is using that to treat people. So this is a remarkably fast, rapid advancement where you've taken the discovery of something that can edit DNA bases at kind of that very resolution of single bases, and now has been used for a variety. And what are the diseases that you see being used, not just at CRISPR, but broadly? Where are we using this gene editing tool for therapies?
So far, I think we found some success in like in terms of disease modifying efficacy. It's been demonstrated only in the past 2 years. It was with the early nucleases, the HIV was it was tried in patients with HIV, But only the past 2 years, we have seen this demonstration of success with clinical trials of CRISPR-based gene editing in sickle cell and beta thalassemia. And, I think but these are all still ex vivo, where, the stem cells are taken out from the patient, they are edited to suppress a particular transcription factor that does not allow a gene to allow the fetal hemoglobin, allows the fetal hemoglobin to express and not shut down. And this then overwhelms the adult gene that produces a mutated dysfunctional protein. So I think this is all still done ex vivo. And it's been successful because the patients so far have not gone back to the hospital, have been, safe, and the treatment has been efficacious. They've not gone through, all the symptoms they were showing because of the sickle cell anemia or for beta thalassemia. So far the data has been good, but aside from that, I think mostly it has been for the monogenic, which is like where a genetic disorder is connected with mutation in one gene. But there's also been tried for diabetes, for cancer, which are like, because of mutations in multiple genes or pathways gone awry. So I think hemoglobinopathies, cancer, diabetes, and what is restricting is the delivery, which is, I think in vivo, they have only been able to try if you saw the recent success of Intellia's delivery of CRISPR to the liver. This was basically a lipid nanoparticle based delivery, but then liver takes up most of the proteins. So it's easy to go in there. Same with retina. It's, you can easily inject in retina, but the target is expanding to many other tissues, skeletal muscles, cerebrospinal fluid, and in addition to retina and liver, and then improving the delivery and the vectors. So I think more and more diseases can be treated once the precision tools and the delivery tools, all these are made better. But right now, yes, hemoglobinopathies and many trials in diabetes and cancer.
Yeah. So again, as you pointed out there, there's a good deal of science of getting this genetic machine to edit the right base pairs, but then it's also the struggle to make sure that it gets to the right cells and into those cells to be able to make those changes so that you can really get some efficacy. I think that there's it's early days, but there's been some great success, particularly in the context of hemoglobinopathies. And we'll get a chance to see what happens. I think in the case of the Intellia work, as you outlined before, is sometimes with a lot of these very cutting edge technologies, oftentimes the goal is to maybe go after a smaller genetic disease for a variety of reasons. One is that there's no unmet need, perhaps. And so you're providing an unmet need for something. And the other one is that usually the biology is more straightforward, and you may be able to kind of make a difference. And I think that once we get past that, sounds like there's lots of other places where we can try to use CRISPR. So
that's the hope.
Yeah, and kind of certainly for those of you that have. I haven't seen the movie Rampage, but apparently CRISPR is even in the theaters and is used in one of these. Jennifer Lopez and I think Dwayne Johnson was in the movie, so it's entered into our zeitgeist the idea of this gene editing. So I'm going to open it up. I am looking for questions from the audience. I think one thing that we could just start with is, where was this CRISPR? Where was this enzyme of gene editing discovered? How did we get our hands on it?
Oh, okay.
Yeah.
So the two or three scientists across France, Canada, and the US, they're working on, they're working in food. I call that industry the food biotech. But anyways, I think they discovered the CRISPR loci in this bacteria that gives flavor to, because there are many strains of bacteria that give flavor to the yogurt, right? And they found this various CRISPR loci in this yogurt bacteria. And they were like, okay, why are these repeat sequences? Because what CRISPR is like, this cluster of repeat palindromic, short palindromic repeats. And it's between that is the spacer of like few nucleotides. They found that these have homology to the viruses that attack bacteria. And so while that discovery was being made, many other bacteriologists and microbiologists, they also reported CRISPR. And it started being used in, more and more information came out of this. And let me tell you that the CRISPR, this kind of discovery is not from bacteria. We have discovered many things from nature. Biotechnology is learning from nature, right? So this is also, CRISPR is learning from nature, because bacteria uses this as the defense mechanism to destroy viruses. Because as soon as the virus attacks them, they are able to tell, oh, I have this CRISPR, between the CRISPR, there's this spacer that actually has homology to this virus. I can create an RNA and have this Cas9 attached to the RNA and go pair up with the viral DNA or RNA and then kill the virus, chop it into pieces. So that's how it was discovered. What Emmanuel Charpentier and Doudna ended up doing was that they actually found the RNA. They were able to report on that RNA that is programmable. And that is how it has become a cost-effective and very tool for us, because you can change the sequence of the RNA and target any DNA, keeping the Cas9 the same.
Yeah, so this modularity, which bacteria actually have already built in to be able to go after their viruses. This was something that the Nobelists, Jennifer Doudna and Emmanuel Charpentier, were able to identify. And now we've seen the movement of that early discovery. Again, I love that this came from yogurt. I mean, for me, this is fantastic, right? People showed enough curiosity to try to understand, like, why was this bacteria growing and not that one, and really dig in. So we can learn a lot. So much of what we've learned from nature. And I think that we didn't go through it, but many of the other delivery methods, like viruses that we use in a therapeutic context, also clearly taken from nature and reformulated for our own use. So I'd say that that's one area that we haven't seen a lot. We've seen a lot of excitement, and I will say that from my read of the field, it's this first gene editing tool called Cas9 has now turned into just like an explosion of identification of these kinds of enzymes. And so, everybody's looking for which one would be the best therapeutic entity, and we don't know yet.
Yeah, there are many Cas9s coming up across different bacteria. And, from bacteria, we have actually obtained so many other enzymes, like in molecular biology lab, all the restriction enzymes we have used for so long, even for PCR, the polymerase, it's all extracted from bacteria. Bacteria found in geysers where enzymes can tolerate high temperature, restriction enzymes that can cut palindromic repeats, and we use in molecular labs to kind of create genetic disease models by cutting at particular DNA sites. And so we have constantly learned from nature, and CRISPR is no different.
Yeah, I'm just monitoring the Q&A here, and one area that came up that you discussed, and maybe we could just spend a little bit of time on that, is the idea of how these gene therapies, we talked about genetic diseases, but what are the ways in which these gene therapies are being used for cancer? And kind of at, again, top line kind of view of that.
Well, right now, some of the approved gene therapies, which are actually cellular gene therapies, are Kymriah, Scarta, Breyanzi, Tecartus, and actually these are all against B-cell lymphomas, which are very, some DLBCL, which are very prevalent. But what they do is basically they add a gene, which is called chimeric antigen receptor, into the cells. So they take the cells from the patients, and which are called autologous cellular gene therapy, where they take the cells from the patients, they add a particular gene that will identify a particular protein only on the cancer cells. And they take the cells, they modify it and put it back into the patients. And these are immune cells from the patients called the T cells. And these cells go find these protein expressing cancer cells and kill them. Similarly, there's attempt to develop allogeneic, which means taking cells from healthy donor cells so that the patients don't have to wait for these chimeric cells to be these modified T cells taken from them to be modified and put back in them. So that wait period is taken off if you have off the shelf CAR T cell, and these are called CAR T cell therapies, chimeric antigen receptor T cells, where you add a new DNA into the T cells, which target the cancer cells. So you make them.
Yeah. Go ahead. Sorry. Yeah. Go ahead.
Yeah. So I've ended my my.
No. I just think that what you described, I think, is kind of a unique element of this, which is one thing was that a gene is not functioning. You put in a new one. Gene is maybe functioning in a way that's negative, so you turn it off. But this one is giving a new instruction to a cell to be able to say, hey, I want you to do something else. And I want you to now go after this tumor cell, and being able to reprogram. I believe the term that, kind of often is used in the language is weaponize the cell, so that now your immune system can go do something that it didn't do before, which is very exciting.
Yeah, I was refraining from using sensational words, but yes, weaponize.
Okay. Yeah, so I mean, we have one question here that I think would be great to kind of maybe connect with is, and I'll just read it out here. Often the public and media have very little scientific literacy, and most don't bother to actually read scientific literature and FDA documents. How can the scientific community be more involved in improving scientific literacy in the media and the public? You want to take a stab at that? That's a big one.
Yeah, well, probably we are doing that right now, a little bit.
I would agree.
That's one step. And I think, there is a lot of limitation about promoting your therapy anywhere by sponsors. So there's some limitation. But yes, science-wise, I think many companies, they actually have, these community labs. I know when I was at Biogen, they had community lab where they would invite students to come in and learn molecular biotechniques and see how these techniques apply to various diseases. And many other companies, they have that. I think that is one way of teaching the young generation how to connect with science, even if they don't take up science, but if they just go and see that in the lab, they're able to understand and get the perspective of how all this applies to treat diseases.
Yeah, so this is, a remarkable thing to be able to bring the young people in, but I think what I might see underneath some of that question is also, the adult population and the scientific literacy and how to be able to bring kind of, these questions around there. I will say that, one of the things that I'm sure that Jaya has spent a lot of time on is those little things that you probably throw away when you get a drug, which is that package insert. That turns out to be, it's not accessible to everybody, but it is a remarkable density of information about the drug. Maybe you can tell us about what goes into that little insert.
Right. Yeah. Well, all of these 1,000 pages of data I was talking about, how, all these data, they go to the reviewers. Actually, that data, the gist of it, the key element of it, gets into these insert packages. which gives you a very high level information about what the dosage would be, how, what were the key clinical trial results, safety, what are the adverse events that the doctor who's prescribing the drug should be wary of, should tell the patients to be wary of. And, what is the limit within which, or even specification about how the drug should be taken. So that is a very important information. It also gives a little bit of small detail about the molecular mechanism of the drug. But I would say that if you go to the website of the company or to the FDA website, not many will go. And I think somebody has just mentioned about scientific literacy. The only way for doing this is, reading I would say a layman version of particular data. The insert package still has a lot of scientific aspects. So maybe, layman people will not be able to understand, but just going and, just talking to your doctor after reading that might make you more aware of what you are actually getting into.
Yeah, it's yet another piece of literature that I'm sure many people don't engage in. So one of the other questions, and I think this gets to your point around how to get young people involved. So there are these lab kind of teaching opportunities. How prevalent are kind of opportunities for students to work in biotechnology companies? I think that internships in academic labs pretty well, that's pretty well established. Lots of people have connections, but does Crispr or other companies that bring in kind of high school or college level students as interns?
Yeah, many companies I've worked for, they do have what I mentioned, community labs, opportunities to work for a week or two during summertime. There are also opportunities where you can, you can go further and actually connect with some of the scientists and see if those but most of the time at high school is mostly limited to a few weeks. But in college level, for sure, there are these what are called the co-ops, and also your own interest in pursuing a scientist and writing to the company for internship. Their internships advertised at the on LinkedIn and on the company website as well. I think CRISPR is planning to start, actually. They're moving to their new site next year, and I think that's a plan. Yes.
Yeah. So the young people, if they think that Facebook is old, then they'll think that LinkedIn is like even for older people. But it is a great place for people to connect and identify people. Mm-hmm. So, Jaya, this has been spectacular. So thank you so much for this kind of educational discussion. I think, I'm hoping that everybody is like starting to finally understand some of those words that come out around all of these clinical trials. And, you provided, I think, a remarkable opportunity to understand how to turn these kind of science into medicine and how challenging it is. Mm-hmm. And, we do want these regulatory agencies looking out for this. So thank you again. I just wanted to plug our next Science Cafe, which is going to be planned for Tuesday, November 30th with Professor Samuel Ting, who is one of our, many Lexingtonian Nobel Prize winners. We're so very lucky to have many in the town, and his Nobel Prize in physics, particularly in particle physics, and he's going to be talking about discovering the cosmos. And if anybody in the audience right now has any ideas about who they would like to see in this venue, I'm going to ask Mary Ellen to put her email into the chat so that people can maybe get a connection there. So we're getting lots of thank yous in the chat. Again, Jaya, thank you so much for your time. This was a wonderful experience. I hope you enjoyed it. I certainly enjoyed it.
Thank you so much, Yogesh. It was an honor coming here. There are a few questions in the chat. Do you think we can handle it?
Yeah. So, one more. So some of these questions are, I think maybe I'm always very wary of going after some of these other things, you know, kind of the Merck pill versus the mRNA vaccine. I think
Yes.
I think what we're going to do is we're going to wait for till we get some subject matter expertise. Scientists are often, you know, wary to venture too far from their home base. But I do hope that we'll be able to bring somebody in who can talk to kind of particularly this lovely new area of biology, you know, kind of what we've learned about the COVID vaccine. And yeah, we're working on it to bring that to Science Cafe near you.
Yes.
All right. Thank you, everybody. Really enjoyed this, and hopefully we'll get a chance to see some of you in person soon.